Chapter 17 Biology Theory Video 15. Now we are going to identify different evolutionary relationships between different species and how they divide or how they are separated from their common ancestors. Now when you go to molecular evidence to compare these DNA sequences, we can actually identify similarities between related species by studying their DNA sequences. Now we know that DNA is mostly found in the nuclei, but DNA are also found in mitochondria and chloroplasts. So all of these genetic information can give us evidence to prove the relationships between species. Now, if the DNA nucleotides if the DNA nucleotide sequences are more similar, that means they are closely related. So that means if they are very similar, that means they have split apart or the separation has taken place very recently. Now if they have less similar DNA, then that means they have split apart long ago. So this is actually common for us and chimpanzees as well. So we say that human shares about 98% of the DNA with chimpanzees. So that means we have had a very close relationship with chimpanzees in a evolutionary term. That doesn't mean that we came from the chimpanzees. So we all have a common ancestor. So now if you compare our DNA sequences with Neanderthal people, then that also we see that we share more DNA sequences than with chimpanzees. That means we have split away from chimpanzees, that is the Homo sapiens have split away from chimpanzees about 1 million years ago. At least 1 million years ago. It's highly likely that it was at least a few million years. But when you compare the DNA of modern human with the Neanderthals DNA, we see that we must have separated, or that separation from Neanderthals must have taken place about 500,000 years ago. Now there is something called human mitochondrial DNA, that is from the mitochondria. This DNA is inherited from the female line, not from the male line. So the zygote would contain mitochondria of the egg but not from the sperm. So as a result, because these mitochondrial DNA are circular and they can't undergo any crossing over, the changes that can happen only by mutation. So because mitochondrial DNA actually show mutations faster than nuclear DNA, it takes about 25,000 years for a single mutation. So different populations of humans have different mitochondrial DNA sequences. So as a result, we can deduce that we have all the Homo sapiens all evolved or originated from Africa and later on migrated to other parts of the world. And also we can safely assume that all of us, all modern humans, whatever the race they came from one single woman known as, we name her mitochondrial Eve. This is not related to creationism. This has nothing to do with it. The mitochondrial Eve did not appear out of thin air. She was a descendant of the earlier human beings. So this is a point of splitting or point of separation. So this date, we arrive at this date, mitochondrial Eve, and this woman must have lived about 150,000 to 200,000 years ago. So from her, the species that evolved from her, started from her, created this present humankind throughout the world. Um, so this date is arrived at. This is a complex process. You need to you don't have to know that in detail, but there is something called the molecular clock hypothesis. So that actually takes some assumptions. It takes the rate of mutations over time, and that the greater the number of differences in the sequence of nucleotides, the longer ago those individuals shared a common ancestor. So then by calibration, by calibrating these nucleotide sequences to follow the time periods, we can, using the fossil evidence, we can figure out that we our this common ancestor, that is a mitochondrial Eve, lived 150 to 200,000 years ago. So now we have learnt about genetic drift and the founder effect, and then we have learnt how these effects would result in the phenotypes being different in different species, and this can be used to explain how the different types of lizards evolved from one group. So they are due to different geographical or whatever the separations, they show different characteristics. So they have different species evolving from the common ancestor. So now in Caribbean to Australia to Cuba, there are different kinds of the same animals found, but they are of different species because they have gone through evolution. So they would have now when you compare some of the animals, when you compare these animals, these lizards from New Zealand and other countries, they would have very common features, but sometimes they are very the primitive features are common. So that suggests that if you go back using a family tree, you will be able to find a common ancestor. So here in this chart on the right hand side, you can see a chart. These are different classes of evolutionary tree. So at the end, in the present day, you have Sphenodon, Agama, Cameleo, Plurus, Anolis, and Heloderma. So these are the Agoma, Cameleo, Opulus, they actually had a common ancestor, and all of them to the leftmost part you have the single ancestor who gave rise to all of these different species by separation. So we can identify these species by using their DNA sequence, by studying their DNA sequences. That gives us the understanding that they have evolved from the single common ancestor. So what results in these animals evolving to produce new species or speciation to occur, that is the separation. As you can see in this map on the left hand side, you can see that these anole lizard species were divided between these small islands. So they got adapted to those conditions, and the different alleles developed in a certain way to make them different from the types of lizards that's found in every other part of the world. But they all have common ancestors. Write down identifying evolutionary relationships. By studying nucleotide sequences of DNA, we can understand the similarities between related species. Not only the DNA in nuclei, but also the mitochondrial and chloroplast DNA can be studied to get the information we need. Leave a line, put a star and write down. The more similar DNA nucleotide sequences are, they are very much closely related and have been separated very recently. Humans are said to have separated from chimpanzees about a few million years ago, and from Neanderthals about 500,000 years ago. By studying the DNA sequences, we can develop a clock known as molecular clock using the molecular clock hypothesis, which gives proof that all humanity are descendants of a single woman called mitochondrial Eve who lived in Africa about 150 to 200,000 years ago. Using the genetic drift and the founder effect, the phenotypes of lizards isolated in different islands of the Caribbean is proof that mitochondrial DNA is related to each species, showing that they have all a common ancestor. Even if you draw this figure 17.25 to make it more familiar, that would be good. Please draw that, and please go through the entire lesson. If you have any questions, please ask. If you don't have any questions, please do the end of chapter questions.